dear students, and a very warm welcome to Eduauraa. This is your biology faculty, Dr. Dhruvi Parekh. So now let us see the distinguishing features of your prokaryotic transcription and the eukaryotic transcription. So let us study the prokaryotic transcription on one side, and we have the eukaryotic transcription on the other side. Okay? Now, tell me one thing. So far, whatever the transcription we have studied, I'm sure you must have understood what is the difference. We are just going to summarize all of them, just clubbing all of them together. So what is the first difference? In a prokaryotic transcription, the synthesis of mRNA occurs in the cytosol, that is in the cytoplasm, because there is no well-defined nucleus. Whereas in the eukaryotic cell, in a eukaryotic transcription, it occurs where? It occurs inside the nucleus, because we very well know the genetic material is inside the nucleus. So even the synthesis of the mRNA is inside the nucleus. Next, again, a very important thing. In your prokaryotic cells, the transcription and translation, both of them can take place simultaneously also. Okay, so that means that along with the transcription, the translation can also be continued simultaneously. Whereas in your eukaryotic cell, in a eukaryotic cell, the translation is always followed by transcription. That means once the transcription is completed, only then you can have the translation carried out, not before that. So you know, in the eukaryotic cell, it is stepwise. That is, first there will be transcription and then there will be the translation. Agreed? Let us take up the next point. The next, we very well know, children, that at the end of your translation, in the prokaryotic cell, what do we get? We get your mRNA at the end, right? Whereas at the end of your eukaryotic transcription, do you get actual mRNA? No, you do not get the actual mRNA. What you get is actually the hnRNA, that is the heteronuclear RNA at the end. Okay, understood this? So that means this is also a major difference, that you are not getting the properly formed RNA at the end of your transcription in the eukaryotes. Again, children, processes like the splicing, capping and tailing are absent in the prokaryotes. Okay, so that means there is no splicing, no capping and no tailing. Whereas in the eukaryotic cell, there is splicing, there is capping and there is tailing, leading to the formation of what? Formation of your ultimate mRNA. Okay? So there is a difference. Again, in your prokaryotes, who is your enzyme responsible for your synthesis of mRNA? That is your RNA polymerase. So here this enzyme in the prokaryotes is RNA polymerase. Whereas in the eukaryote, this enzyme is particularly RNA polymerase 2. Okay? This enzyme is particularly of the RNA polymerase 2 type in your eukaryotic organisms. So these are all the distinguishing things between your prokaryotic transcription and eukaryotic transcription. Remember, children, the end product of transcription has to be your mRNA, the messenger RNA. Understood this transcription? So this is your biology faculty, Dr. Dhruvi, signing off for now. But I'll be seeing you guys in the next session with some more interesting topics, interesting concepts. Till then, keep learning, keep watching Eduauraa. Thank you.